Okay, so we have two chapters. We have chapter 8, which was cellular division. And we have chapter 9, which was genetics. Which of those two chapters would you prefer that I go over first? Any preference?
No.
No preference at all? Okay, we'll start with chapter 8, and we'll talk about cell division. So remember that there are two different forms of cellular division that we talked about in the lecture, and that was mitosis, which is the division of somatic cells, and meiosis, which is the division of reproductive cells. Remember that somatic cells are any non-reproductive cells. So liver cells, muscle cells, kidney cells, brain cells, any of those types of cells are going to be considered somatic cells. Reproductive cells are going to be your germline cells that give rise to your sperm and or egg. So what I'd like to do is draw again that chart that we had before to compare and contrast the similarities and differences between these two. And then what I'd like to try doing is building a flow chart that will allow you guys easy access to determining if the cell that you're looking at on the exam is undergoing mitosis or meiosis. Okay, so let's go ahead and just start with the general table regarding mitosis and meiosis. Alright, so my table. We've got mitosis, meiosis, and then this is going to be characteristics. Okay. So our first is going to be the one we've already discussed. That's going to be the cell type that performs these different forms of division. The cell type for mitosis is going to be somatic cells. For meiosis, it's going to be reproductive cells. Okay, after cell type, let's talk about the number of rounds of division. How many rounds occur in mitosis?
2.
Mitosis. Just one, right? You go through once and then you divide. So it's 1 round of division. What about meiosis? 2. In meiosis, we've got 2 rounds of division. Okay? So first big difference there. Mitosis just goes through 1 round of division. Meiosis goes through 2. Okay? Now to get to the answer I think you were getting towards, and that is number of cells produced. How many cells are produced at the end of mitosis?
2.
2, and by the end of meiosis?
4.
4, right? Okay, good. So, 1 round of division for mitosis gives us 2 cells. 2 rounds for meiosis gives us 4 cells. Next, let's talk about
Chromosome sets.
yes. Let's talk about the chromosome sets. Okay. At the end of mitosis, the cells have how many chromosome sets? Parental. 2, right? So it's going to be 2N, which allows us to classify those cells as diploids. Okay? What about at the end of meiosis? 1 chromosome set, which means they're going to be classified as haploid cells. Okay? Then, let's do genetic content. In other words, the DNA of those daughter cells. Is it identical or is it diverse? So with mitosis, by the time we produced our daughter cells, if we look at the DNA in each, is it going to be identical or is it going to be different between those daughter cells? It's going to be identical, right? Okay? What about at the end of meiosis?
Diverse.
It's going to be diverse. Good. Okay? And finally, let's talk about the role that each of these processes play. Mitosis is going to be used in things like growth, development, and wound healing. Meiosis is just going to be used in the production of reproductive cells. All right, so this is our chart, our table for this process. Excuse me. Now what I'd like to do is take some of these concepts and present you with a general idea in regards to flow charts, so that on the exam you can ask yourself some very basic questions and start to eliminate one of these two different types. So for instance, if I give you a cell and I say, is this cell performing mitosis versus meiosis, what are visual cues that you can look for that will let you know one way or another? Okay? So first and foremost, let's start with the easiest one. Are 4 cells produced? And that's the easiest way to determine one versus the other. If the answer is yes, you're dealing with meiosis. If the answer is no, you can't immediately say that you're dealing with mitosis, because you might just be in meiosis 1. If the answer is no, there's another question you have to ask. And that question should be, are the sister chromatids one color or 2? Okay? So are they one color or are they 2? If all of the sister chromatids are 1, then you're dealing with mitosis. If, however, there are 2 colors, red and blue or something along those lines, then you're dealing with meiosis. So if I give you a cell or a collection of cells that have divided, then you have to ask yourself, are you dealing with 2 differently colored sister chromatids or homologous chromosomes? Okay? And if you're not, then you're dealing with mitosis. If you are, you're dealing with meiosis. There's no reason for you to have 2 different colors if we're just talking about mitosis, because all we're looking at is the division of the cell. Okay?
How will we know, will we know if 4 cells are produced in the question?
I will tell you, or you'll just see the picture of 4 daughter cells being produced.
Okay.
Okay? Yeah, I will tell you if 4 daughter cells were produced. So just count the number of cells. If it's under 4, you're going to have to go with this side of the flow chart. But if there are 4 cells shown, or I tell you that there's 4 cells produced, then that lets you know, hey, you're dealing with meiosis. Okay? All right, so that's the first place you should start. Start with the easiest stuff. 4 cells is a dead giveaway, so start with that. If you can't go from there, then you'll use the rest of this. Okay? Any questions about this flow chart? Because we're going to do another now in just a second. Okay, the next flow chart that we're going to talk about. Okay. Are the daughter cells haploid? So if I at any point in time mention that they're haploid or they're 1N, and the answer is yes, then you are immediately dealing with meiosis. The answer is no, you're dealing with mitosis. Now, if the daughter cells have not yet been produced, if we're, say, in metaphase, and I haven't produced any daughter cells, you can't really answer this. Because remember, up until Anaphase 1, all the way through Anaphase 1 until Telophase 1, in meiosis, we'll still be dealing with diploid cells. So at that point in time, you should be looking for either different colored chromosomes, red and blue versus purple, or other visual cues that will let you know you're dealing with mitosis versus meiosis. Okay? All right. Any questions about this? Next. Question you should ask is, what is happening in metaphase? And when I say that, what I mean is, tetrads or homologous chromosomes are lining up. If that's the case, then you're dealing with meiosis. Okay? So if you see those tetrads or those homologous chromosomes lining up at the center of the cell, then you're dealing with meiosis. The other option is sister chromatids. Now, again, we can't immediately say that if it's sister chromatids lining up that we're dealing with mitosis, because remember, in meiosis 2, the sister chromatids also line up at the center of the cell. So we have one more series of questions to ask. Are those sister chromatids the same color or different colors? If they're the same color, we're dealing with mitosis. If you see them have a little bit of a different color, then you're dealing with meiosis. Okay, so this is looking at metaphase. Obviously, you have to be able to identify metaphase to know that you're looking at it, but understand that this is the difference and discrepancies that you should be looking for. Okay? The next question, or the next statement that you should really kind of pay attention to. This is going to come to somatic versus reproductive cells. And this is going to have to deal with particular buzzwords that you'll hear. If I'm talking about somatic cells, so how do you know if the cells are somatic? So that's the question you want to answer. Okay? So there are two pathways. Again, first off, if you hear the word reproductive, sperm, egg, anything like that, then you're not looking at somatic cells. Instead, these are reproductive, and that means you're going to be looking at meiosis. So if you hear the terms reproductive, sperm, or egg in regards to cells dividing to produce that, then you're dealing with somatic. Okay? If, however, you hear things like liver, or skin, or muscle, or brain, or any other kind of cells that are not reproductive cells, then you're dealing with somatic cells. Now there's one final thing that I want to talk about here before I draw the final box to say somatic and all that. And that is a fertilized egg. If you are told at any point in time that the sperm and the egg have combined, thus fertilizing the egg, you're going to then be dealing with what's known as a zygote, and a zygote is 2N, and it is somatic, or composed of somatic cells, which means you're going to be performing mitosis. So understand, if you're just looking at the production of sperm, the production of egg, or the division of reproductive cells, then yes, you're dealing with meiosis. But once that fertilization event happens, you're no longer dealing with meiosis. You are officially in mitosis. So this means that you're dealing with somatic cells, and that means you're dealing with mitosis. Okay. All right. Any other questions about stuff like that? Okay. I think the last thing I want to talk about from this chapter, outside of obviously the fact that you should know what all of the stages of mitosis and meiosis look like. And those, I think, are pretty easy to memorize if you just look at the pictures in the book. You know, there's distinct things happening at each stage. And meiosis looks pretty darn similar to mitosis. So usually what I'll do on an exam is I will pull one of the figures from the book, and I'll ask you questions about it, whether it's mitosis or meiosis we're looking at, as well as what stage of this is it. And I think from pro-metaphase on, it's a really, really easy job to pick out. I think interphase and prophase are typically the ones that kind of trip students up. For those two, just remember, look at the nucleus. If the nucleus is nice and uniform in color, it's interphase. If, however, you see those pairs of sister chromatids starting to form, and I think in the book they look something like this. Okay? Once you see the DNA condensing like that in those pairs of sister chromatids, you're no longer looking at interphase, you're looking at prophase. Okay? Yeah, you're no longer looking at interphase, you're looking at prophase. Because interphase, the DNA has not condensed into these structures yet. Okay. Are there any questions over the crossover event, or homologous chromosomes versus sister chromatids, or non-disjunction events? Because we did talk about non-disjunctions at one point in time. Any questions over those? Would y'all like me to go over any of that in particular? Let's do non-disjunctions. I think that's a good place to start. Okay?
If you know the things that will be at the exam, why not go ahead and...
Yeah? So just go over what's going to be on the exam?
Exactly. That's the benefit.
Okay. Well, all of it's going to be on the exam, so. Okay, well, let's talk about non-disjunctions. It's like my favorite answer. Okay, what non-disjunctions are is, you'll remember in meiosis, we're doing 2 steps. In meiosis 1, we're dividing homologous chromosomes while in meiosis 2, we're dividing sister chromatids. Non-disjunctions occur when we have difficulty with this or this. So there's a non-disjunction that can happen in meiosis 1, and there's a non-disjunction that can happen in meiosis 2. So what I'd like to talk about is the % chance that you'll wind up getting some form of sex cell, either sperm or an egg, that would still be usable, that would not result in kind of any aberrant effects, as we saw with like the trisomy of chromosome 21. So let's start with the nondisjunction in meiosis 1. So I'm going to draw a cell up here. We're going to follow it through meiosis 1 and meiosis 2, and we're going to see eventually what happens. Okay, so here's my cell. This is going to be it during metaphase 1. Okay, so remember we have those homologous chromosomes that are paired up. They've got that crossover event happening. Looks like this. And then it's got another set of chromosomes that look like this. So they're my homologous chromosomes. I've got my spindle structures formed. They're ready to pull this apart. Now, in a nondisjunction of meiosis 1, one of the sets of homologous chromosomes, one of the tetrads, will not effectively be pulled apart. So during anaphase 1, these two here are going to be divided like we would normally expect, but this one here is going to be pulled all to one side. So once I go through anaphase 1 and telophase, when I have my two new daughter cells, what I'm going to see happen is this daughter cell here is going to wind up with a pair of sister chromatids that look like this. It's got a little bit of blue. So that happened normally, the way we would expect. But then I'm going to have both of these homologous chromosomes, which means they're going to look more like this. And they probably have crossed over. It's going to look like this, and it's going to look like this. So I now have technically 3 pairs of sister chromatids in this cell. If I come over here, I'm going to find that I only have 1 pair of sister chromatids that looks like that. So when I perform metaphase on these cells, this is the event that's going to happen. Okay, so this, I guess just so you can see, this is the end of meiosis 1. And here is meiosis 2. So that when each of these cells then divides, here is the overall outcome in terms of my final cells. So I'll have a nucleus, each of these cells. In this cell here, my nucleus will look like this. I have 1 long red, 1 short blue, and then 1 short red with a little bit of blue. In my next one, I'm going to have a long red with a little bit of blue on it. I'm going to have a short blue with a little bit of red on it. And then I'm going to have a short red. Okay. Over here, though, I'm just going to have this. And this. So my DNA content for each of those cells is going to be, this one is going to be N plus 1 because I have 1 extra chromatid, or chromosome in this case. This one's going to be N plus 1. This one's going to be N minus 1. And this one's going to be N minus 1. So, what is the percent of cells with the correct amount of DNA at the end of a nondisjunction of meiosis 1? Do any of these have the correct amount of DNA? No, we're expecting these cells to just be 1N. These two are 1N plus 1. These two are 1N plus, or I'm sorry, minus 1. So that means we have 0%. So that means if any of these cells become a sperm or an egg, the offspring resulting from those cells has no chance of having the correct number of chromosomes. Everybody sees that? If it gets either one of these, it's going to have 1 extra chromosome. If it gets either one of these, it's going to be short 1 chromosome. Okay? So that's a nondisjunction. in meiosis 1. Let's look at a non-disjunction now in meiosis 2. So you can see these two compared side by side. Right? So here we go. Non-disjunction in meiosis 2. Try and draw this exactly the same as I did the last one so that we can see the difference between the two. Okay, so what this means is first and foremost, when I perform meiosis 1, my crossover event, I'm going to see the following. I'm going to see the correct and appropriate division of my tetrads. So that means when I get to the end of meiosis 1, I get to my two cells that are formed. When I get into meiosis 2, I'm going to see the appropriate number of pairs of sister chromatids in each of my cells. So this one here is going to look like this. This one here will look like this. And when I come over here, I'm going to see this. And then over here, I'm going to see this. Okay? Now, the non-disjunction is happening here in meiosis 2. So what we're going to see happen is in one of my cells, the spindle structures are going to appropriately attach to my pairs of sister chromatids. In the other, though, one pair of sister chromatids is only going to be attached on one side. When that happens, I get the following as the output. So here are my 4 daughter cells. At least reasonably similar in shape and size. Okay. So, nuclear envelope reforms. Okay, in this first one, what I'll see is something that looks like this. In the second one, I'll see something that looks like this. In my third one over here, I'm going to see something that looks like this. Okay? And then in my final one, I'm going to see something that looks like this. So, if I classify these cells based on their genetic content, each of these first two is classified as 1N, or just N. But these last two are where I'll see the discrepancy. This one is N minus 1. This one is N plus 1. So, in regards to the same question, percent of cells with the correct amount of DNA, what's my percentage of cells that wind up with the correct amount? 50%, right? So, understand that if the non-disjunction happens sooner rather than later, we're going to wind up with 0%, whereas if it happens in meiosis 2, then we're going to wind up with 50%. So here, this is the non-disjunction. While here, this is the non-disjunction. And those are the differences and discrepancies between those two. Questions, concerns?
Is it always 50% for meiosis too?
Yes, and you'll see the reason why is because you have one cell that correctly divided here and one cell that correctly divided here. So this was fine. Meiosis 1 happened correctly. The only time this event is happening in this example here is in this cell. This cell's unaffected by it, and that's why you get 2 that are correct and 2 that are not.
Okay.
Now, this is an independent event, which means, in theory, both of these cells could be undergoing a non-disjunction at the same time.
Okay.
But the likelihood of that happening is far lower. So that's why you're almost always going to come out with 2 cells that are good, 2 cells that are not. Whereas here, it doesn't matter what happens independently in these 2 cells, the damage has already been done up here.